A cyclotron is, think of it kind of as a subatomic particle raceway, and it's a way for us to accelerate protons, smash them into a target, and have that target change from one element into the other. Well, what happens is when these decay inside the body, they actually produce a signal. And a special detector that we have around the patient is able to detect where that signal came from in the patient. By back mapping each of those events, we're able to construct a computer map of the distribution of the tracer in the body. We take advantage of the fact that tumors tend to accumulate more of the radioactive material than the normal tissues around them, and therefore tumors tend to look like hot spots showing up on a background of lower activity in the organs. Radiopharmaceuticals, by their very nature, when they're used in nuclear medicine, are decaying away, similar to uranium or plutonium or any of the other isotopes that people read about in the papers. But what we're going to be looking at with this are tracers that decay with relatively short half-lives. And so fluorine will decay with a half-life of about 110 minutes. Choline-11 will decay with a half-life of about 20 minutes. And that means if you start out with 10 units of a material, for instance with C-11 Choline, after 20 minutes you're only going to have half of that left. It's actually going to decay away into another daughter element. We'll have a wider palette of available options here, but it will benefit our patients because we're able to apply those to research projects that were already in progress in Mayo, and we're able to carry those forward down here as a secondary site for Mayo Clinic. The future is very bright for PET. I think in the past couple of years we've seen huge growth in the number of indications where PET has been found useful. In addition, unlike other types of radiology tests, such as CT, for instance, since we keep having different flavors of contrast materials, and we call them tracers, but they're the radiopharmaceuticals, we are developing more and more, and Mayo has a very active development program putting together syntheses for these new novel tracers. We're able to come up with different types of tracers to address different types of cancers. Well, I think in the future there's going to be a very big emphasis on targeting specific types of chemotherapy, immunotherapy, radiation therapy to specific types of patients, depending upon what their gene profile is and what their molecular profile is. And I think what we're going to be doing is helping to identify and stratify those patients into the appropriate bins, if you will. And so if we can get them into the appropriate bins, then we can use that information in order to provide more targeted therapies to those affected patients.